My presentation is mutagenic properties of artificial sweeteners. Sweeteners are additives that are dispelled into foods to give sweetness that is similar to that of table sugar while having none of the associated calories. Artificial sweeteners are used on a near daily basis, especially for people diagnosed with diabetic diseases. The common use of these chemicals brings into question of whether the sweeteners are hazardous or dangerous to consume, especially considering that some major brands, like saccharin, have received carcinogenic classifications from Congress and the FDA in the past. Research into these chemicals is vital to correcting the already dire health conditions many Americans face in their everyday lives. To elaborate on early concerns, Congress passed a 1977 act that required saccharin to have a cancer warning label on its associated products due to studies on rats correlating its consumption to the development of tumors. FDA members Gene Taylor, Morris Weinberger, and Leo Friedman published a journal describing findings on saccharin causing tumors, specifically bladder neoplasms, in rats. In the study, 5 out of the 11 total tumor cases were malignant cancer tumors. In addition, out of the 3 rat groups, which were each fed different amounts of saccharin in their diets, the group that consumed the greatest amount of saccharin developed 9 out of the 11 total diagnoses of bladder neoplasms. Findings like these are what caused immense concern over the safety of artificial sweeteners and are what fuels the need to research the effects that sweeteners have on people who consume them in great quantities. In the experiment, the gap of research is that few studies have been conducted on the mutagenic effects of artificial sweeteners like sucralose. The question this experiment will answer is, does sucralose or saccharin cause mutations in DNA? And the proposed hypothesis is, if K12 E. coli is grown in different medias containing saccharin and sucralose, then the artificial sweeteners will induce mutations in the DNA of bacteria when compared to the control variable. The scientific procedures that will be carried out are to first receive the agar slant K12 E. coli. Then, the broth mixtures that are needed to grow the bacteria will be created by mixing sterile, deionized water, luria broth, and artificial sweeteners. Then the broths are inoculated, which consists of scraping the bacteria slant, then swirling the scooping loop into the broth mixture. The broth will then be placed in the shaking incubator where it will grow for 2 days at 37 degrees Fahrenheit at 100 RPM. Then the samples will be transferred to 1.5 ml tubes and centrifuged into pellets of cells. After these procedures, DNA will be extracted from the pellets, which includes lysing, precipitating, and ethanol washing the cell pellet. Finally, the DNA will be run through a gel electrophoresis test, where results will be analyzed to determine whether visible mutations occurred in the base pairs. To help with these lengthy procedures, Marcus Stafford, a molecular geneticist, taught me how to carry out many of the procedures while keeping a sterile environment. The procedure I am about to start is running the gel electrophoresis analysis, which will take only a couple hours. These procedures will be carried out on the 5th of December 2023. After these results are achieved, I will collect the data by analyzing the gel electrophoresis results and looking at the bands to determine whether genetic mutations have occurred. If the band locations of the sweetener groups are different than that of the positive control, then the artificial sweeteners cause mutations to the K12 E. coli bacteria. An unexpected outcome of experimentation that occurred during early phases of the procedures was when it was determined that the E. coli in the 2% saccharin group was growing at a much slower rate than the rest of the groups.